Can you guys see my screen?
Yes.
Okay. Well, thanks for the nice introduction. Okay, so today my talk is going to be a little bit, I'm going to do a little bit of a divergence into the how I ended up where I'm at on my career path, because this is the Fellows Symposium, and I guess you're supposed to do that sort of thing. And then I'll talk about the machine learning work that we have been doing, which kind of creates sort of a cohesive story over the last several years. I'll spend a little bit more time talking about some of our newer work on sets of partial genomes and making predictions with incomplete data. And then finally, at the end of the talk, I'll talk about some of the curation work we've been doing in order to fuel these efforts with the time that I have at the end. Excuse me, back in, I was an IGB Fellow back from 2010 to 2013. I was actually Carl's last postdoc. He passed away at the end of 2012. And it was really quite an exciting experience. It was also quite difficult, you know, when he passed. But I spent a lot of time with Carl talking about evolution and, you know, the things that he was interested in and working on projects with him. And one of the things that people don't really appreciate about Woese is that he studied evolution and he thought about evolution as signatures. And by signatures, in this case, I mean nucleotide sequences, subsequences of the 16S ribosomal RNA that either established relationships between related organisms or sort of demonstrated that organisms were less related. And so if you go back through Woese's old papers, you'll actually see, you won't see trees. I mean, you don't start seeing trees until a little bit later. You see these tables of relationships, and that's how he actually discovered the archaea and as a separate kingdom. And those are all basically K-mer analyses using the T1 digests that he kept in old grade books in his desk. And when I was working with him, we had a couple of different projects going, but one of the ones that we actually drove to completion was an analysis of the evolution of the Mycoplasma group. They're actually descended from a set of organisms that have a wall, but they had been classified into a phylum of wall-less organisms, and it didn't appreciate the fast clock nature of their evolution and stuff like that. And so I was building trees and making alignments for Carl, and he was getting frustrated with me because, you know, I was a postdoc. And one day he finally just rolled out a big poster of the 16S, and he pointed to it and he said, "That one, that stem loop right here is the difference. This is why this is the signature that demonstrates that the Mycoplasma are related to the Erysipelotrichia, those are the walled organisms that are similar to them." And I couldn't believe it. After all of those years, 30 years later, he could remember every stem loop and every signature within this molecule. So that kind of blew my mind, but it was cool. So after he passed away, I ended up going to Argonne National Laboratory. Argonne is a US Department of Energy lab, and it has a fairly diverse and large scientific portfolio. Most people know us for supercomputing and the APS facility where people go to get protein crystal structures. So if you're into protein structure work, you know Argonne. There's a large portfolio of materials in energy science work, climate science work, and there's a fairly robust life sciences set of life sciences people and portfolio. The entire laboratory is administered by the University of Chicago. So we all have, most of us have joint appointments to the University of Chicago and Argonne National Lab. When I moved to Argonne, I started working on this project called PATRIC, and PATRIC stands for Pathosystems Resource Integration Center. This is a NIAID-supported bioinformatics resource center It's the PI is Rick Stevens. He's the associate lab director. And the PATRIC is an umbrella project that contains, a lot of different tools and stuff, but probably the most famous tool that it contains is RAST, which stands for Rapid Annotation Using Subsystem Technology. So if you're into bacterial or archaeal genome annotation, you're probably familiar with that tool. And the database underneath of RAST is called the SEED. So all of this is bundled up in the PATRIC project. And I started working on those resources when I went up there. More recently, we've recompeted that contract, and we've merged with Viper and IRD, the Viper and IRD bioinformatic resource centers, which are dedicated to viral, supporting viral work. And that sort of a super group with JCVI now. And we're also on a trajectory to merge with the people who run the eukaryotic bacterial resource centers, which are UPathDB and VectorBase. So the PATRIC is a large database of approximately 500,000 genomes with web tools for comparative analysis and sort of services that are fairly computationally expensive or have a lot of moving parts like assembly and annotation. And originally, the group that developed a lot of these services, that's from the University of Chicago, Argonne, and the Fellowship for Interpretation of Genomes, they specialized in predicting and projecting protein functions of core machinery, so metabolism, and the core functions. And one of the reasons is because they were really dedicated to sort of the basic science of characterizing microbial life. But around 2014, NIAID really started to push us on building up a capability for studying antimicrobial resistance. And we were actually we are a pathogen resource, so that made a lot of sense. So we developed this sort of twofold strategy. One was to sort of just get the annotations right for antimicrobial resistance, which is non-trivial. Some of the antimicrobial resistance genes are really, really well characterized, down to like single nucleotides, and some were not. And so we've been straightening those out and getting them able to project in protein space. And was done largely by Gary Olson, Veronica Vonstein, and Svetlana Gerdes. And also providing the linkages back to the other resources that are really important for studying antibiotic resistance, like CARD and the NCBI NDARO database. One of the thing that I did, I kind of took a left turn, and I started trying to figure out how to predict antimicrobial resistance phenotypes and genomic regions algorithmically. That was a little bit different of a problem. So if you have a genome and you want to know if the genome, if the organism that had that genome was resistant or susceptible to a given antibiotic, most people just look for the AMR genes and they say, well, I found a beta-lactam, a beta-lactamase, so it must be resistant to beta-lactam antibiotics, and that actually works pretty well. And there are good public resources for doing that. But this is where I sort of took a deep dive into machine learning. I didn't know what machine learning was at the time. This is like, I don't know, 2014. And I started doing some of my own tinkering with building my own algorithms, and then sort of finally got serious and realized that this whole universe of algorithms existed. And so it is possible to use machine learning algorithms to make genotype to phenotype predictions, and the cancer literature existed at this time, where people were doing a lot of work. This has some advantages over gene-based methods. You don't need to know the gene content or the SNP patterns, and you don't need reference databases of the SNPs or the genes. And so it has the really nice property of not needing much curation in terms of genes and SNPs, but it does require genomes with good clean phenotypes in order to make these assertions. And then the machine learning models are neat because they can capture complex genetic relationships that are hard to identify even with genome-wide association studies. So they have kind of a lot of potential for use as a discovery tool. Excuse me. I also had this long-term goal, which I still have, but and that is to try to, contribute to reducing clinical response times. When it comes to um, prescribing antibiotics and determining whether an antibiotic will work before prescribing it, we're still in the dark ages. And, the clinical gold standard culturing usually takes at least 36 to 48 hours for phenotyping fast growers like E. coli and Klebsiella, for instance. And it literally requires enrichment and plating and growing the organism in the presence of an antibiotic. I had really been jazzed about point of care sequencing as an AMR phenotype diagnostic. My position on that has become a little bit more nuanced, and you'll see that towards the end of the talk. But sequencing is one potential way to provide a speed up that could potentially save lives, and so I was interested in that. So at the time we got into this, there were a few papers that existed on predicting antimicrobial resistance phenotypes and genomic regions from genome sequence data, but there weren't many. And since then, a fairly large literature has sprung up around this, that's, not just our work. So without going into the gory details of how, the machine learning models work, this is basically how you build one. If you want to build a machine learning model for predicting a discrete phenotype like susceptible or resistant, what you do is you build a classifier where you take susceptible genomes and resistant genomes, and you apply the machine learning model to those in order to, and the machine learning model, the classifier, finds the things that are indicative of susceptible and indicative of resistant. And it takes these sort of salient features so that when you have a new genome, it looks at the new genome and it literally votes in order to make the prediction. They're really fancy voting schemes. That's really all there is to them. These can become arbitrarily complex. You can do susceptible, intermediate, resistant, and there's an entire universe of algorithms under the hood. But and there are different features you can choose, like nucleotide chimeras or proteins or amino acid sequences and stuff like that. But basically, it's an algorithm that's searching for the difference between, a set. That's what a classifier is. And in our work, we found that the ensemble methods work really well for predicting phenotypes. So AdaBoost was the one I first glommed onto. And since then, we've been using a lot of random forest and XGBoost models. Other groups have used other models to good success. So set cover machines and support vector machines are pretty common. And we're starting to see more papers using deep learning methods as well. I have stayed away from those because it's harder to figure out what the models are learning sometimes. But there seems to be sort of this interest in those, and we're starting to see incremental improvement in those. There are disadvantages to predicting, to using machine learning models in this way. They require a lot of genomes. They require good antimicrobial susceptibility test data, and limitations in the diversity, the sampling, the geographic sampling, phylogenetic diversity, the accuracy of the susceptibility tests, variations in susceptibility tests can all bleed over into creating error in the models or making the models not generalizable and stuff like that. So there's a lot of considerations for doing this, including also making sure that your sets are balanced and that you're not overfitting and stuff like that. And so we've published papers building classifiers. The first one I think I published in 2016. And we actually have some classifiers in RAST right now that probably need to be updated. But they're, so that's all out there. But we wanted to challenge ourselves. And Rick Stevens said, you should just try to get, predict minimum inhibitory concentrations. And I said to him that I didn't think that it was possible. And then we did it. So I was wrong. But you can predict minimum inhibitory concentrations as well. And the way you do that is the same as building a classifier, except that you're building a regression model where you're attempting to predict a number along a range. And so there's not much difference there. So the current workflows that we use in the models that we have, we start with assembled genomes. To get a little bit more detail here, you can build them from reads. I don't like to, but they work. And we start with cleaned metadata. The assembled genome knocks down the size of the input considerably. And then what we normally do is we use features that are k-mers. Nucleotide subsequences that are typically from 7 to 15 nucleotides long. Smaller nucleotides give you sort of frequencies, like distributions, and longer k-mers give you more unique features. So if you're trying to look up regions of the genome, you go with a longer k-mer. If they get too long, your memory footprint gets too high. If they get too small, the accuracy goes down. But they're anything from 7 to 15 works pretty well. So we take the counts or the frequencies of those k-mers. We one-hot encode the antibiotic names. So basically assign a 0 or 1 digit string for the antibiotic, and then the label, which is either susceptible, intermediate, resistant, which is one-hot encoded, or the minimum inhibitory concentration. Then we apply the machine learning algorithm to this matrix, and that's what gives us the model that we use to make predictions for a genome of unknown phenotype. We usually keep all of our antibiotics in one model, but in a lot of our papers, we separated them just to show, the difference in effect. If you keep all the antibiotics together, it does the model will do a little better. So in 2018, we published 2 papers sort of laying out how to do this. And it's sort of one body of work, but it's 2 different studies. The first paper that we wrote on this was a collaboration with Jim Musser's group at Houston Methodist Hospital. They had a large collection of Klebsiella pneumoniae genomes that had the standard laboratory panel tests, and with the BD Phoenix machine. And I think there were about 1,700 genomes in that paper. The second paper we wrote on this was, and this one was far more detailed. This was on Salmonella, and it was a collaboration with Pat McDermott's group at the FDA. Pat is the head of the NARMS program, and that stands for National Antimicrobial Resistance Monitoring System. And they track Salmonella and other outbreak strains like Listeria, Campylobacter, Shigella, some of the Shigellas. And it's sort of an umbrella program that's a collaboration between the FDA, USDA, CDC, and state and public health agencies. That particular collection at the time had 5,287 genomes, Salmonella genomes, non-typhoidal Salmonella genomes, with minimum inhibitory concentration data collected over 7 years. And the samples were collected in surveillance mode. So there's like a random element to the collection. So it was a nice collection because it had good coverage of susceptible strains, and it was sort of a little bit less biased by these extreme resistance phenotypes that encourage sequencing at hospitals. So these are the results for that model. The box plot shows the accuracy for each antibiotic, the median accuracy on a 10-fold cross-validation is the red line. The edges of the whiskers are the first and fourth quartile. And I think that the box is the 95% confidence interval on the model. Now, and the accuracy on these MIC prediction models is bounded within plus or minus 1 2-fold dilution step, because that's the limit of resolution on most of the automated antimicrobial susceptibility testing machines. So, if you were within 1, we declare victory if we're within 1 dilution step. So this was pretty good. The overall accuracy of this model was around 95%. And you can see that there's differences among the antibiotics and so forth, but I was surprised by how well that did. We had to downsample the genomes in order to get them into memory, which was also kind of interesting. So this model had 4,500 genomes in it. And the heat map here, the average accuracies kind of dust over what's actually happening with the accuracy for each minimum inhibitory concentration. So over here in the heat map on the right, we have the antibiotics on the x-axis here, the y-axis, and then the x-axis is obviously our minimum inhibitory concentration. And the color of each cell is the accuracy within plus or minus 1 twofold dilution step for that antibiotic MIC combination. And the numbers are the number of genomes for each of those. And what we found is that for the most part, the accuracy tends to track with the number of genomes that we have for a particular bin. Because the Salmonella data set was so evenly, so well collected and curated, we were actually able to do some other fun things, like looking for biases by serotype, phylogeny, state of isolation, collection year, and contamination source. And we didn't find any. But down here on the bottom, I have the map of the United States, and that's the accuracy by state. There are different, the number in each state is the number of genomes in the collection from that given state. So that also tracks with the depth of sampling a little bit. And then people kept asking us, how many genomes do we need in order to make a good model? And so we finally did that. This plot here in the top, the red line is the accuracy within plus or minus 1 twofold dilution step for a model using the 250 most diverse genomes, the 500 most diverse genomes, the 1,000 most diverse genomes, and so on. And the dashed line is the 95% confidence interval. And what this shows is that, as we add genomes, we also gain in accuracy, but it's a little bit misleading because the genomes out here at the end, we're losing diversity as we're adding them. So it's a little bit more optimistic than I think it actually should be. So the whole genome sequencing based ML work, it works well for classifying susceptible and immediate and resistant phenotypes, and it's fairly straightforward. There are other ways to do it besides nucleotide chimeras, which I've talked about and people have published on. Predicting MICs is possible. The minimum inhibitory concentrations, that's possible, but it's harder to get the data for those. The accuracies, as the field progresses, the accuracies will improve incrementally with better methods. So as people apply deep learning methods and better sampling, we're going to get better and better outcomes. In the Salmonella work, we had maybe half of the genomes had sort of a diagnostic quality accuracy and error rate. And those will continue to improve. And we may actually end up seeing diagnostics that are sequence-based like this. But the models depend heavily on the quality of the input data. So after we did those, when we were doing this work, we stumbled across a fairly interesting thing, because Marcus Wen did this work, and he and I were sitting there talking about the models one day, and basically, one of the criticisms of this is basically, well, if you just built this and it just found the antibiotic resistance genes, and so you haven't learned anything. And we didn't think that was in our gut instinct, but it was a good criticism of the work. So we went back and we did an experiment where we built a model using only antibiotic resistance genes. So this heat map right here is the result for Klebsiella pneumoniae, 1,700 genomes from the Houston Methodist Project. So it's the same thing I showed before, with only for Klebsiella. And then we threw out everything but the well-annotated antibiotic resistance genes. And what you see here, if you're trying to look between each heat map, is that there's not much difference. There's a few places where we get these little subtle differences in the accuracy. The lighter green is the most accurate here, but they're nearly identical. And I said, you know what, Marcus, go ahead and throw away the AMR genes and leave everything else. What happens then? Well, the answer comes back as it's pretty much identical. So we learned from this experiment in this paper that you don't need the antibiotic resistance genes in order to make a reasonable prediction about the phenotype. And this blew our minds for a little bit, but then we started thinking about it and we were like, you know what? Maybe this is finding the bits and pieces of the plasmids that we didn't throw away. You know, maybe it's finding unannotated genes, or there's got to be a reason for this. So we decided to, take the lid off of this and look at it a lot more closely. So we designed an experiment where we used core genes that are held in common among the members of a species and are not annotated as being involved in AMR in order to try to predict antimicrobial resistance phenotypes. So we built models from sets of these core genes. And what I'll show you is data for 100 randomly selected core non-antimicrobial resistance genes. And we drew sets of these randomly. So we repeated it 10 times and built models from the set of 100 genes. And then we compared those models to models built from whole genomes. And we did this for Salmonella, Mycobacterium, Klebsiella, and Staph. And what we found when we did this is that the models are very similar. The 100 conserved gene set model is the orange bar, and that's the average of 10 sets of 100 and the error bar is the minimum and maximum 95% confidence interval. The blue bar is the accuracy, the F1 score for a single model built from the whole genome for the same organisms. And so these accuracies track pretty close, or these F1 scores and the accuracies track pretty closely, and the error rates, which I'm not showing, also track very closely. And so what we find is that an antibiotic that had a low accuracy for the whole genome also has a low accuracy on one of these randomly selected set of core gene models, and vice versa. So we were struggling to understand this, but basically we kept drawing samples and seeing very little variance across samples. We found in general that the accuracies tend to be 5 to 10% lower than whole genome models whenever you're building models from sets of core genes. The error rates are slightly higher, obviously, because they correspond with the accuracy. And we also observed a similar observation when we went to predict minimum inhibitory concentrations. And we did a lot of control experiments to try to show that it's not the result of overfitting, memorizing of labels, algorithm choice. We switched out algorithms, k-mers as features. We did alignments instead of k-mers and SNPs. And so we did a whole bunch of things. And we ended up publishing that in October in PLOS CompBio. And that was kind of cool because PLOS CompBio offers a full reproducibility peer review. And so we signed up for that. So someone downloaded the GitHub repo and actually repeated everything to make sure that it worked. So I thought that was kind of cool. So the next thing that we started working on, and this work is work that was done by Daria Aiton, who was a visiting scholar with me from the Technical University of Denmark. We wanted to look more closely over the entire genome in order to understand the regions of the genome that had good antimicrobial prediction signal and phenotype prediction signal, and those that had bad prediction signal, because the ones that have very good prediction signal should have some sort of correlation or relationship to AMR. So in this experiment, we used Klebsiella, Mycobacterium, and Salmonella. And the Klebsiella data set's about 1,700 genomes. Mycobacterium was 6,000 genomes, but it was downsampled to about 3,000. And the Salmonella data set was about 5,000, but it was downsampled to about 2,500 because we couldn't use them all. And for these, what we did is we aligned all of the Klebsiella, all of the Mycobacterium, and all of the Salmonella against a corresponding reference strain. So Salmonella was aligned against LT2, Mycobacterium, all of those genomes were aligned against H37RV. And it was only a chromosomal alignment. We threw away the plasmids and the plasmids and extra chromosomal stuff. So then we had these large, full chromosomal alignments against the reference. And what we did is we chopped those up into randomly selected subalignments, covering the whole chromosome of chromosomal alignment of different lengths. And Daria built random forest models to predict susceptibility versus resistant phenotypes from subalignments of various lengths. And what we see down here, whenever we look at those, this plot shows the area under the receiver operating characteristic curve, which is basically a proxy for accuracy. What we see with these is that as the alignment length goes up to about 200 kb to maybe 300 kb, the accuracies begin to increase dramatically, and then they begin to taper off. And once we get out to about 300 kb, we see the same thing that Marcus saw, which was that we get models with a reasonable amount of accuracy, but they're 5 to 10% lower on average than a whole genome model would be. But now that we had these, we had the ability to look across the genome at which regions had predictive power. And so this set of plots shows the model performance by location. So we took, for Klebsiella and Salmonella, 5 kb subalignments and built models from that subaligned region, and then plotted the AUCs for those. And what you see is actually a general pattern of reasonably good accuracy, all the way across for these. But that there's some regions, especially in tuberculosis, where you see these really dramatic peaks of high accuracy. Those turn out to be RPOB, INH, isoniazid, one of the isoniazid resistance genes, ERM-A and ERM-B. So those are antibiotic resistance genes. And then we see these valleys that were a little bit more difficult to explain, but whenever we started looking at what was there, we realized that these were phage elements in areas of the genome that are poorly conserved. And so we have less nucleotide conservation in those regions. And so there's nothing to learn. So these are phage elements. And another thing that I would point out from these that I thought was really fascinating is this beautiful banding pattern of colors based on the antibiotics. So up here at the top of this chart, we have ciprofloxacin, and levofloxacin. So the fluoroquinolones are up here with high accuracy. And then down here at the bottom, we've got trimethoprim sulfamethoxazole. And so these different antibiotics have a different footprint across the genome. They have the accuracy that can be drawn from them is sort of a proxy for how many changes or how many, correlated changes have occurred in the genomes of these. And so there's a really interesting tie back to the epistatic effects of the genomes. And it's a little bit muddier here in tuberculosis, but because those genomes are much more closely related. So you can see it over here in Salmonella as well. So we were interested in that in particular. So the partial genome alignments can be used to identify signatures of antimicrobial resistance, and but we wanted to look more closely at these, especially for signatures that spanned a larger phylogenetic distance. So what we did is we did an experiment where we separated the strains of each species based on camera distance. We used this as a proxy for percent identity, so it's a proxy for relatedness. And then we established a clustering threshold, and we clustered members at a given clustering threshold. And if two genomes were members of a cluster, they were only allowed to appear in either the training or the testing set of a 5-fold cross-validation. And the reason this is important is because we didn't want to train a model on something that was, and then have it see something that it was similar to whenever you went to test the model. And so what that means is that if a pattern then is found in the training set, it has to be a more strongly conserved pattern in order for it to be seen in the testing set. So it's forcing the model to learn patterns that are more phylogenetically conserved over a larger distance. And so we started plotting these and looking for, trying to understand models. So what we did is any sub-alignment that had an accuracy over 80%, we kept it for further downstream analysis. And at various and we looked at this at various clustering thresholds and so forth, but I'm showing you the 99% clustering threshold for Salmonella right now. And what Daria did is she went and she colored the genes based off of whether they had an annotation to one of the well-known databases for transporters, virulence factors like VFDB, AMR genes using PATRIC and CARD, or whether there was no hit to be found among those. And what we find is that a lot of these correlated regions are involved, have an AMR related function or a virulence related function, because virulence and antimicrobial resistance track together. And there's a lot of transporter related matches here. The y-axis here is the number of antibiotics for which we had an accuracy above 80%. So there's some here that, and I think this is because of related classes of antibiotics and being tested. But so what we're finding is that there are a lot of regions that have good predictive power in the genome that are related to virulence and transport, whether the transport was actually annotated as being involved in AMR. But we also found a bunch that had no hit. And a lot of these turned out to be metabolic genes. And that was a little bit of a dead end for us because I don't have a wet lab, but we're working on trying to understand some of these. But there was a recent paper by the Collins group. I think they're at MIT. It was in Science, and they were looking for genes that were involved in antibiotic resistance, or the genes that were involved in antibiotic resistance among metabolic genes that were, basically hidden in the metabolism of the genome. And some of the matches that we found corresponded with what they found, even though we were looking at different organisms. So I have some confidence that we've been doing this every time we write a paper where we're trying to figure out what the genes mean and what the models mean, and we're getting better at this. And so we're starting to get down to lists of genes that need to be characterized. And that's on the bleeding edge. The Daria's work was submitted to M Systems, and we've had a full round of peer review on it, and I'm feeling pretty good about our chances with them, although you never know. But I'm optimistic. I got fingers crossed. And so the gene lists and all of that work should be available soon. So we have ongoing work. The ongoing interest that I have is that I like these models as a useful tool for identifying genomic regions involved or correlated with antimicrobial resistance. And I like them as an inroad to beginning to understand the sort of more complex epistatic effects of various antibiotics over the evolutionary trajectory of the organisms. I also like this as a framework for making assertions about partial genomes, because a metagenome is basically almost always a collection of partial genomes. And so, if you can make a prediction for a bin, a metagenomic bin, like how well can you make a prediction for a metagenomic bin? And so I think that there's a future for building tools that bioinformatic tools for making assertions based off of what you have in hand rather than what you would have had if you had a whole genome. And I think that also makes more sense from a diagnostic perspective, but I don't know if we'll ever get an accuracy that's high enough. So I would qualify that statement. We're doing a fair bit of grant writing and sort of laboratory work to try to design bench experiments to verify the models. The Argonne has become a lot more interested in these robotic lab capabilities, kind of like the iBioFab talk that we saw this morning. And I think that actually our group has talked with those guys. I have a small amount of funding from a DARPA project for doing some machine learning work, and we've been building models for predicting other features. I mean, not to beat the drum over and over, but it is possible to predict host organisms, like what organism was the bacterium isolated from. Some organisms are easier than others. Salmonella is fairly straightforward, but E. coli is a little harder. It depends on, it's very metadata dependent. Environment of isolation isn't too bad. It's usually pretty easy to tell the difference between hospital strains and environmental isolates. Human body site is hard. Predicting what human body site an organism came from is hard because organisms are typically generalists, but there is some signal for some organisms, so that's kind of neat.
Did we lose Jim?
Can you guys hear me?
Yeah, now I can. I think we lost you for a little bit there.
Okay. Well, I'll start back up here where I was talking about models for predictive features. Sorry about that. But basically what I was saying is that it's possible to build models for a variety of different bacterial metadata categories, including what organism it was isolated from, the environment that it came from, and that we've spent a fair amount of time looking at growth conditions, like trying to collect biolog data to predict genomic regions involved in growth on a given substrate. And the DARPA project that we're part of is trying to distinguish between a pathogenic and non-pathogenic species. And so we've been looking at host cell transcriptional responses, because the way that the device for this project is being designed is that there's a eukaryotic reporter cell that will fluoresce in the presence of given pathogens. And we've been trying to come up with differential fluorescence patterns. So we're pretty close to submitting a manuscript on that. And then finally, gene content, because we want to know kind of what genes ought to be there and so forth. So, and a lot of this stuff, I mean, I think back about, my time with Carl Woese, and he wasn't particularly interested academically in antimicrobial resistance. I think he kind of felt like it was sort of it was important, but it wasn't, the main goal was to characterize microbial life. And so, I don't think that he would have had that much interest in the AMR work that I've done, but I think that if Carl were still alive, he would be very interested in the use of machine learning algorithms as a form of signature analysis. The machine learning algorithms are sort of like a well worked out way to do signature analysis, as long as you can design good experiments and understand their output in biology. And I think that he would have been fascinated by these and the outputs that they provide, and also that he would have been very, very keen to see how far he could push these in order to work out the early patterns and the evolution of life. So all along, in doing all of this work, I've been sort of restricted because I've needed the phenotypes and the genomes in order to build the models. And so all along the way, I've been carrying a large data curation effort in order to fuel this. So it's somewhat selfish, but it's also under the auspices of providing a resource for the public. So whenever somebody sequences a genome and then publishes a paper, they have to submit the genomes to NCBI or somewhere else. But antimicrobial susceptibility test data and other metadata like host organisms and environments and things like that is not compulsory. So our group has been collecting susceptibility test data from the literature, collaborators, NIAID projects for years. And we keep them all on an FTP site on the PATRIC website. And I've finally written a small review article about this that's in peer review right now. The collections, I would say it's modest by the standards of the number of genomes that are out there today, but we currently have 67,000 genomes with some form of susceptibility test data, whether it's susceptible, intermediate, resistant calls, or minimum inhibitory concentrations. Got 128, at least 128 antibiotics in the collection. Most of them are the clinical antibiotics that we all know, and 89 species. So, and over 200 publications that have been sort of mined for this data. If you look at the genomes in the database right now that have some form of AMR data, there's about, this is capped at 500, but the more purple ones are more genomes here. It's genomes that we've seen before, although I would say for the purposes of this that some of the more obscure organisms are starting to fill in a little better, and some of the tracking organisms like Campylobacter, the Campylobacters are starting to actually, you know, come up on the map here with enough genomes to be useful. If you take the collection as of November and you drive it down to the number of organisms that you could actually make classifiers for predicting susceptibility and resistance, this is what you end up with. And if you read down the left column here, this is the number of genomes again. The color is the number of genomes again. If you read down the left column, it's basically the main pathogens where antibiotic resistance is an issue.
Jim, sorry, I don't think we can see your slides since you came back. Sorry about that.
Let me see if I can fix this. Why am I having trouble?
Last one of the day, and
I know, it's like Zoom got tired. Okay. How about now?
there we go.
Hey, sorry about that. So I was just showing that this is the sort of the total distribution of genomes with susceptible and resistant calls in the PATRIC database right now that we've curated so far, with purple being the most deep sampling and the lighter white being less sampling. And if you reduce that down to the ones where you have at least 50 susceptible and resistant genomes, this is kind of like what you can build models off of. And 50 is almost too small to build a model. But and the, you know, what's across the left here is what's sort of clinically relevant as for antimicrobial resistance. For minimum inhibitory concentrations, the data are more sparse, in part because they're spread over more, you know, bins. But we have data for Acinetobacter, the Campylobacters, E. coli, Klebsiella, staph, and strep, or strep and Salmonella, and a little bit on Pseudomonas, a little bit on Mycobacterium, and, you know, a little bit on Enterobacter, which is growing, but I don't expect much from Mycobacterium. Within the database, there's also, there are these really profound geographic and funding related biases in the data collection. So the top 4 organisms right now with antimicrobial susceptibility data and genomes are tuberculosis, gonorrhea, Salmonella, and strep. And for tuberculosis, the deepest sampling, that's the dark purples and stuff, are coming out of Asia, Africa, and South America, as you would expect, because those are places where tuberculosis is endemic. But it's not evenly sampled over the different countries. It's being sampled in countries where there's basically a sampling capacity. But then if you go over here and look at Neisseria, you know, gonorrhea is being sequenced and sampled and sequenced in the US, Canada, and the UK, basically. There's some data from Japan and stuff. And then Salmonella is something that the US is very much fixated on, except for the Indian subcontinent where you get a lot of typhi data and paratyphi data. And then strep pneumo, we got, you know, the US and China pretty much. So these are important because for a lot of organisms, you can see there are large differences in the geographic sampling and that are due to clinical practices and a variety of other factors. Using the data that's in the database, we've actually built a large model for each for the predicting susceptible and resistant phenotypes and predicting minimum inhibitory concentrations. The average on this model was about F1 score is about 90%. And you can see this is the accuracy on the green plot. And I showed the purple one before. And then the leftmost and the top rows are the row and column are the average. And then this is the accuracy of our most recent minimum inhibitory concentration model. It's a little overly optimistic because we have some pretty light sampling in some of these bins, but it's showing progress. And I think that's the important thing to note is that our ability to make predictions and to understand some of this stuff is growing as the data grow. So finally, with the last minute or so, I guess you're not really a microbiologist if you haven't been impacted by COVID. And I'm not a primary data generator and I'm not a clinician. So the way that I've been helping to sort of support the effort is that my old buddies at Houston Methodist Hospital pulled me in, and as part of the BRC, I've been helping them to assemble and analyze the genomes that they're sequencing. And this group that I'm working with has sequenced over 50,000 COVID genomes. So for a period of time, I think this effort was the largest sequencing effort in the United States. I think that the CDC Spheres Consortium, some of the people there may have sequenced more at this point, but for a while there, we were really cranking them out. So with that, I'd like to thank the BRC team, the DARPA Friend or Foe team, which is the lead institution on that is Texas A&M, our Houston Methodist collaborators, our FDA collaborators. Marcus Wen, he's the one on the left here. He did most of the modeling work. Daria, who's second from the right, did all of the work on the subalignments. Margot Vinofelin, who isn't pictured, did a lot of the metadata curation that has helped fuel these efforts. And we have funding from NIAID, the NIAID Bioinformatic Resource Center program and DARPA, the DARPA Friend or Foe program. With that, I can take questions.